Welcome to the official channel of Woodbine Modern School. It's your biology class of class 12th. It's lecture number 3 of human reproduction. Our today's topic is gametogenesis. Gametes means what? The gamete term means the one cell which will fuse to form zygote. And genesis means formation. So gametogenesis simply means formation of gametes. There are the primary sex organs that the male and female has are respectively testis and ovaries. So testis in males, they produce what? They produce sperms, and ovaries in female, they produce what? Ovum. So these two gametes are being produced and the process is known as gametogenesis. So in the case of male, immature germ cell known as spermatogonia are produced, which will further leads to the formation of what? Sperms. And this process begins from puberty. The spermatogonia, or the singular form is what? Spermatogonium. This is present where? This is present in the seminiferous tubules. Present where? Inside the wall of seminiferous tubules, and they multiply by meiotic division. Meiotic division, the term meiotic division means what? The equational division or the equal division, and it will cause the increase in number. Each spermatogonia is diploid and contains 46 chromosomes. So each spermatogonium means what? Each spermatogonium will lead to the formation of a diploid one containing what? 46 chromosomes. Students, diploid means what? Diploid and haploid, these two terms are being used for denoting the number of chromosomes. Haploid means what? If a organism has X, or if a cell has an X number of chromosome, that means it's haploid. And if it has a 2X, yani double of that chromosome number, that means it's a diploid one. So the spermatogonium is what? Spermatogonium is diploid, yani twice the number of chromosome. That means if the haploid contains 23 chromosome, the diploid will contain how much? 46 chromosomes. Now in this figure, we will understand where the spermatozoas are being produced. This is the spermatogonium layer. These are the Sertoli cells. These are the primary spermatocyte, and from there there's a formation of secondary spermatocyte. This secondary spermatocyte will lead to the formation of spermatids, and spermatids will further mature to form what? Spermatozoa. This spermatozoa is also known as sperm. So the events are being occurred in this way, that the primary spermatocytes periodically undergo now meiosis. Why? Because ultimately the sperm have to maintain haploid one. Why? Because X number of chromosomes will go from sperm and X number of chromosomes will go from ovum. Both will fuse to form a diploid organism. Okay. So to maintain the number of chromosome, this meiotic division is very much important. So primary spermatocytes must go under first meiotic division so that it could reduce the chromosome number. So that when the organism will form final organism, it will only contain 46 number of chromosomes. Otherwise, if this meiotic division will not occur, then the person may have 4x number of chromosome, which is an abnormality. We must have only 2x. So 1x will come from the male and 1x will come from female. The secondary spermatocyte undergo the second meiotic division. The second meiotic division always is similar from the mitosis. So mitosis is also the equational one, and the meiotic division, the second meiotic division is also the equation. Means what? The chromosome number remains same. It will not reduce. So to produce 4 equal haploid spermatids, the spermatids transform into spermatozoa or sperms by the process called spermiogenesis. So the formation until the spermatids, they are known as what? Spermatogenesis. And the formation of spermatozoa, the formation of spermatozoa from spermatid is known as spermiogenesis. So there is one term called spermatogenesis and the other term is known as spermiogenesis. So after spermiogenesis, sperm heads become embedded in the Sertoli cells and are finally being released from the seminiferous tubules, and this process is known as spermiation. So they're released from the seminiferous tubules known as spermiation. FSH acts on Sertoli cells and stimulates secretion of some factors which help in the process of spermiogenesis. So FSH is what? Follicular stimulating hormone. FSH stands for follicular stimulating hormone, and it acts where? It acts on Sertoli cells and it stimulates the secretion of some factors that helps in the process of what? Process of spermiogenesis. That helps in the process of spermiogenesis. Now, here comes the structure of a sperm. We will look into the detailed structure of a typical sperm. So here the sperm has which kind of parts? We will look into it. So first is there's a presence of plasma membrane. It is compulsory because sperm is also a cell, so it must have plasma membrane. Now if we try and divide the whole sperm part, then we can divide it in a head part, then it has a neck part, then sorry students, so it has what? It has the head part, then it has a neck part, and then it has what? The middle piece. This is known as, and this part is known as tail. So the head part, neck region, then we have what? Middle piece, then we have what? Tail of the sperm. So the head part, first we'll look into the head region. So this head part has what? Plasma membrane. Then there is a bulged region, you can see. This is known as what? Acrosome. This acrosome is very important. Why? This is a fluid filled membrane, and this fluid which is filled here, this acrosome, is very important for the fertilization process. Now the nucleus, this nucleus contains what? This nucleus contains chromosomal material. You know that nucleus contains what? Chromosomal material means the X part. Now this is the neck region. Now comes to the middle piece. This middle piece, you can see the zigzag material. This zigzag material is what? This is nothing but mitochondria. This mitochondria is very important. Why? Because it provides energy to the sperm to travel from the male genital area to the female genital area. So this mitochondria is very much important. And now this is the tail part which helps to swim through the female genital tract. This tail part does what? It helps the sperm to swim easily into the female genital tract. So these are the details which I have told you regarding the structure of sperm, which I have listed down. The red regions are the important one, which I have highlighted with the red part, so you can read it. Now, the sperm released from the seminiferous tubules are transferred by the accessory ducts. I have already mentioned you the process of the transformation of sperms from testis to the vas deferens, and then how it ejaculates. So the seminal plasma along with the sperm constitute the semen. So semen constitutes what? Seminal plasma and sperm both. And the function of male accessory ducts and glands are maintained by what? Androgens. Androgen is the male hormone which is required for these activities. Now we will come to the Now we will start with the female gametogenesis part. So this is the structure of what? This is the structure of ovary, a typical ovary. So we will see that there is a lining of blood vessels, which is very important. Each and every organ, whatever constitute the human body, have this blood vessels which is required. And there is what? A primary follicle. Follicles are what? Follicles are the layers which surrounds the primary oocyte or the main cells. So, the primary follicle are what? The cells which surrounds the primary oocyte. Now, the primary follicles will mature gradually to secondary follicles. Now, this secondary follicle will further forms what? Tertiary follicle, having what? The antrum region. Antrum region is what? Antrum region is nothing but it's a fluid filled space which is present with the primary oocyte. This primary oocyte will gradually leads to the development of graphene follicle. Graphene follicle is nothing but it constitutes the more mature part of the tertiary follicle, that is the theca externa. The layers are called theca externa and the inside layer of the graphene follicle is known as theca interna. Now this primary oocyte will gradually leads to what? Leads to the development of secondary oocyte. The secondary oocyte will gradually leads to the formation of ovum and first polar body. First polar body means the polar body is the dead part, means this polar body will detach from this ovum and this ovum will be get matured. Now after the release of ovum, this graphene follicle gradually leads to the formation of corpus luteum. If this ovum gets fertilized, this corpus luteum will maintain its structure and it will produce progesterone, which is important for pregnancy. And if this ovum does not get fertilized and menstruation starts, this will leads to the formation of corpus albigans. So you can see in this structure, this graphene follicle will lead to what? Corpus albicans. Corpus albicans forms when this ovum will not fertilize, and corpus luteum will form and produce and maintain its structure when the ovum will get fertilized. So the process of formation of mature female gamete is called what? Oogenesis. Oogenesis initiates when? At the embryonic development stage. And the gamete mother cell, this oogonia, as you have seen that in case of male it's spermatogonia, in case of female it's oogonia. So oogonia is formed within the each fetal ovary and no more oogonia are formed after birth. This is very much important to know that the viability in case of males exist, means what? They can produce sperms onto there throughout the life, but for female there is a fixed number of oogonia which are being produced at the time of birth only. After birth no more oogonias are formed. So these cells starts division and enter into the prophase 1. In the prophase 1 get arrested and the oocyte which is present is known as primary oocyte. So in this figure you can see that the primary follicle and having the primary oocyte, it stops over here in the ovary at the prophase 1 stage. The release of ovum, this the whole stage starts at the puberty. And this is called primary oocyte and primary follicle I have already discussed. So at the puberty only 60,000 to 80,000 primary follicles are left in each and ovary, and new theca interna are produced when there is a formation of secondary follicles, as I have mentioned. And antrum, I have mentioned that it's a fluid filled cavity which is present with the secondary follicle. Theca layer I have discussed, there are two theca layers, inner and outer, and they will form the graphene follicle or the tertiary one. An unequal division resulting in the formation of haploid secondary oocyte, or you can say the ovum and the first polar body. So no, the secondary oocyte and the tiny first polar body first is produced, and after that the second polar body releases when the ovum is produced. So the secondary oocyte retains bulk of the nutrient rich cytoplasm. So this question is given in your textbook, that can you think of any advantage for this? Advantage for what? Advantage that the secondary oocyte retains bulk of the nutrient rich cytoplasm of the primary oocyte. So the cytoplasm of primary oocyte have bulk of nutrient in it. Why? Because in case of humans, the male consumes its all nutrition for reaching to the female genital tract. So when it comes to the female genital tract, it doesn't have much, the cytoplasm or the nutrients are being already consumed, but the zygote needs that. That's why, to compensate the loss for the male part, the female part, or you can say the ovum or the secondary oocyte, contains this bulk of nutrients. So you got the answer, that what is the advantage of this bulk of the nutrient rich? To just compensate the loss of the sperm nutrition. Does the polar body born out of the first meiotic division further degenerate? Actually, the some says that it may divide, or the some textbook says that it may not divide. So this question is not very much clear till date. The tertiary follicle further changes into mature or graphene follicle. The mature follicle or graphene follicle, and the layer is what? Zona pellucida, which surrounds the secondary oocyte. Now, the difference between the spermatogenesis and oogenesis is what? That in spermatogenesis, throughout the life of a male, the spermatogenesis occurs and the sperm gets produced. But in case of oogenesis, you can see that oogenesis have the oogonia cells limited at the time of birth. No more oogonia are being produced, but sperms are produced. And the other major difference between spermatogenesis and oogenesis is what? The division. In spermatogenesis, 4 male sperms are produced, but in case of oogenesis, only single ovum is produced. So with this particular figure or diagram, you will be very much clear that how these things are worked. So we will see part by part. So first is spermatogonia, this primary sperm cell. The primary sperm cell, we will first, I would like to divide this whole diagram or the whole figure in 3 parts. First is this event of the cells which is going through the stages of the human life, and then the third part is what? The number of chromosomes which is being divided over here. So in case of male, first we will see that what? First spermatogonia is there. Spermatogonia will produce what? Primary spermatocytes through mitosis. So chromosomal number will remain same because this is a mitosis division. So in mitosis, the chromosome number remains the same. The 46 means 46. Primary spermatocytes will form and having the first meiotic division. First meiotic division is occurring. So 46 will convert into 23. And the formation of spermatids is there. So after the formation of spermatids, spermatozoa is produced with the process of differentiation. Here no division occurs, just the circular cells will come into this shape, the tail one. So this is here in case of male. Now in case of females, oogonia is here, as spermatogonia is here, here oogonia, mitosis differentiation occurs. So from single cell, primary oocytes are formed. Here primary oocytes will continue to form, and at the time of birth it will cease at prophase 1. After that, puberty occurs and first polar body releases, and there is a formation of secondary oocyte. Further, there will be a release of what? Ovum and second polar body. So now this second polar body can degenerate or it can further multiply. This question is still unknown, students. Now there is a formation of ovum. So this chart I think is very much clear to you. If you have any doubt, let me know in the comment section. Please try and draw this chart on your own so that you can know that how the chromosome numbers are being divided and how the stages are being divided. So this is a very important chart which I have put in your slides, and it's there in your textbook. Just try and mug up this whole chart. So this is all for today's lecture. Thank you so much.